Now we will go to another interesting talk by Professor Abdulla Alangari, who is a professor of pediatrics at the College of Medicine, King Saud University. He is also the president of the Saudi Asthma and Immunology Society. Professor Abdullah did his pediatric residency at the King Fahad National Guard Hospital and Massachusetts General Hospital before completing his fellowship in allergy and immunology at the Harvard Medical School. His talk today entitled Genetics of Pediatric Inborn Errors of Immunity. You are heartily welcome, Professor Abdullah. The floor is yours.
Thank you very much. And thank you for the organizing committee for inviting me to this wonderful conference. I hope my voice is clear and you can see my slides.
Yes, we can.
Okay, thank you. Certainly the elegant presentation of Dr. Mahawij have made it much easier for me to proceed with my presentation. My talk, as you see, is about genetics of inborn errors of immunity, a general overview and personal experience. I will, try to take you with examples from the publications of special patients that I have contributed to. You've just heard that inborn errors of immunity are a heterogeneous group of genetically determined disorders of the immune system. They're characterized by at least one of the following: increased susceptibility to infections, which we call primary immune deficiency, autoimmunity, autoinflammation, atopy, and/or increased risk of malignancy. Just to quickly refresh your memory about the components of the immune system, it's mainly composed of two major parts, the innate and adaptive. The innate is composed of physiological barriers, phagocytic cells like neutrophils, macrophages, NK cells, complement, cytokines, while the specific or adaptive component is composed of a humoral part, which is a function of B lymphocytes, which produce immunoglobulins, and cellular part, which is a function of T lymphocytes. As you also have heard, the identification of more diseases of inborn errors, genetic disorders of inborn errors of immunity have started to increase significantly from the mid-90s, but after the introduction of next generation sequencing, there was a marked surge in the identification of inborn errors of genetic causes of inborn errors of immunity. 1 year or more, 1.5 years ago, that they were about 450, and now they're approaching 500 different genes affecting every component of the immune system. The first clear description of inborn immunity was the famous Proton's agammaglobulinemia, which is an X-linked disease with a description of a patient who had serious bacterial infection that was corrected with immunoglobulin infusion in 1952. In general, the discoveries of different genetic defects of the immune system over the years have greatly advanced our understanding of the immune system and how it functions. For example, defect of FOXP3, which is a transcription factor necessary for the development of regulatory T cell, leads to a disorder called IPEX, immune dysregulation, polyendochronopathy, enteropathy X-linked, which is a defect in peripheral tolerance. And the discovery have enhanced our understanding of the role of regulatory T cells and peripheral tolerance in general. Also, the discovery of defects in AIR gene, which lead to a disorder called autoimmune glandular syndrome type 1, which is the defect of central tolerance. And this also have greatly enhanced our understanding of central tolerance in the thymus. As in many other genetic systemic disorders, we see great variability in clinical features of inborn errors of immunity, even in patients with identical genetic variants. Inborn error of immunities are inherited as autosomal recessive, which is the most common form, or autosomal dominant, which can be loss of function in the form of haploinsufficiency or dominant negative effect, or gain of function, and less frequently X-linked. Also, different genetic defects may have the same phenotype because either they are involved in the same pathway, like for example, the common gamma chain defect, which is an X-linked disease, and JAK3 deficiency, which is an autosomal recessive disease, both causes KID-like picture, or SCID phenotype, and they are in the same pathway, so they present with the same phenotype. Also, GP91 and P67 deficiency both cause chronic granulomatous disease because they are components of NADPH oxidase enzyme, which in which defect lead to CGD. GP91 is encoded is an X-linked defect, is an X-linked disease, while P67 is autosomal recessive. On the other hand, the opposite side of the coin, one gene defect may result in different modes of inheritance and phenotypes. For example, different defects in CARD11 gene can be inherited as autosomal recessive, which presents as KID-like, or autosomal dominant with loss of function, which can mainly present with severe atopy, or autosomal dominant as gain of function, which present as hypogammaglobulinemia. Other mechanisms of genetic defects that lead to inborn error of immunity are somatic mutations affecting different populations of cells. They are very infrequent and easily overlooked. Examples include defects in FAS gene, which leads to autoimmune lymphoproliferative disease called ALPS, or NLRP3, leading to neonatal onset multisystem inflammatory disorder, NOMID. Those diseases, phenotypes are usually similar to diseases resulting from germline defects in those genes. The first case example, this is one of my patients who have a homozygous mutation in purine nucleoside phosphorylase PMP gene, leading to a defect in this purine nucleoside phosphorylase enzyme. This enzyme is very important in purine metabolism pathway, which ended up in uric acid production. Defect in this gene lead to accumulation of substrates that are very toxic to the cell and lead to defect, especially of lymphocyte proliferation, presenting with severe combined immune deficiency. Those patients have low T cell number, as you can see here. CD3 is very low, very poor T cell function in comparison to control, and the level of PMP enzyme would be, or enzyme activity actually, is very low compared to normal control. This figure is of an E. coli transfected with the mutated gene versus wild type gene. Well, this is the Western blot showing expression of normal expression of the wild type protein versus the defective or mutated gene in that patient. Another example, rather than severe combined immune deficiency, this is combined immune deficiency. This is a family that we're following that were diagnosed with IKBKB mutation. This gene codes for inhibitor of kappa kinase beta deficiency, or results in deficiency of this protein, which is very important in the NF-kappa-B activation pathway, and its deficiency leads to defect in NF-kappa-B function. As you know, NF-kappa-B is a very important transcription factor in the function of many cells in the body, especially in the white cells, and lymphocytes are particularly affected in these cases. And those patients present with infection, wide range of infection to multiple organisms, including BCGitis, CMV, systemic CMV, infections in the pneumonitis, and recurrent also viral, other viral and bacterial gram-negative and gram-positive infection in the form of sepsis, meningitis, and so forth. Those patients again present with also hypogammaglobulinemia and poor T cell function, and interestingly, many of them, like in those in this family, have delayed separation of the umbilical cord, similar to patients with leukocyte adhesion defect. So those patients need bone marrow transplantation, obviously, otherwise they would not usually survive beyond the first or second years of life. Another example of a defect leading to B cell hypogammaglobulinemia, B cell defects, and also NK and cytotoxic T cell defect, is the X-linked lymphoproliferative disease. So this patient was a 5-year-old boy who presented with recurrent pneumonias and and hypogammaglobulinemia, and found to have a defect in SH2D1A gene, which codes for SAP protein, which is important for B cell activity, as well as NK and cytotoxic T cell activity and ability to kill target cell virally infected cells. This patient also developed lymphoma as a complication of this disorder, and those patients also susceptible to developing hemophagocytic lymphohistiocytosis and fulminant infectious mononucleosis secondary to EBV infection. So these are disorders that affect primary immune deficiency disorders, affect immune defense, and present with susceptibility to broader range of organisms. But some PIDs have disorders have restricted infection susceptibility. For example, those genetic defects affecting what's called pattern recognition receptors, which are part of the innate immune system and recognize common pathogenic ligands. For example, toll-like receptors, which are about 9 or 10. They are pattern recognition receptors that can recognize viral RNA or DNA, bacterial lipopolysaccharides, etc. Defects in TLR3, for example, usually lead to poor response to some viruses with low type 1 interferons, meaning interferon alpha or beta, and subsequently poor defense against viral infection and severe viral infections. This diagram or figure shows here TLR3 as intracytoplasmic receptor responding or activated by double-stranded RNA, and activation leading to downstream activation of several proteins, eventually leading to activation of IRF7, interferon regulatory factor 7 and 3, leading to upregulation of type 1 interferons, which are released by the infected cell and bind to its receptor on certain leukocytes, such as macrophages or T cells, leading to also activating this pathway that was discussed before by Dr. Mahawij, and which involves JAK1, type 2, STAT1 and 2 IRF9, and also leading to further increase in expression of type 1 interferons and interferon stimulated genes, which are important for defense against viruses. This pathway, one of the important regulators or down regulators of this pathway is a protein called USP18, which inhibits this pathway at the site of JAK1, and ISG15 is important to stabilize USP18. This patient, which was a 2-year-old Saudi boy with recurrent herpes simplex encephalitis, who we discovered in collaboration with Jonra Casanova group to have a TRF homozygous mutation in TRIF, TRIF, which is downstream of TLR3, and led to a deficiency and a very poor response to TLR3 and 4 ligands, leading to very low type 1 interferon and interleukin-6 production. Interestingly, as you know, herpes simplex virus is a DNA rather than RNA virus. So we don't understand why there's a special increased susceptibility to this kind of virus. This is an example of this of the effect in this pathway. Also, other examples include reported patients with IRF7 and IRF9. If you go back here, IRF7 and IRF9 defects lead to increased susceptibility to severe influenza viral infections with influenza pneumonia, severe influenza pneumonia. While on the other hand, defects in JAK1, STAT1, 2, or IRF9 do not usually or are not associated with increased susceptibility to influenza, but are associated with increased susceptibility to other viral, severe other viral infections. This is usually a demonstration of what's called the redundancy phenomena, which is basically the idea of this is that some infectious organisms defense depends on multiple or the multiple pathways involved. When there is a defect in one pathway, the other pathway can adjust for that. Now we switch gears from primary immune deficiency diseases to other forms of conditions called autoinflammatory disorders, which are inborn error of immunity resulting from dysregulated innate immune response with lack of autoreactive B or T cells. So they are not autoimmune disorders. There are different types. One of the major types is so-called inflammasomopathies, which result from dysregulated interleukin-1 beta activation. A classic example is monogenic periodic fever syndromes, such as familial Mediterranean fever. Another major group is that we will discuss in more details is interferonopathies, which result from increased production or lack of inhibition of type 1 interferons. And there are other pathways involved in autoinflammatory disorders as well. So this is just to demonstrate that those type 1 interferon pathway is a very complex and is activated by presence of double-stranded DNA in the cytoplasm or viral genomic material in the cytoplasm, leading to increased production of type 1 interferon eventually from the infected cell or involved cell, which binds to its receptor in the leukocytes, leading to activation of the pathways discussed before and upregulation of interferon-stimulated genes. So this is a patient that we recently reported, which had a mutation of in USP18. USP18, as I said, is an important regulator of type 1 interferon pathway activation. It acts by displacing JAK1 from its binding site to interferon alpha receptor 2. If it is completely absent, this results in a very severe disease with death in late gestation or early infancy. If it is mutated but not completely absent, as the case of the patient I will discuss, it results in very severe disease, including hydrocephalus, severe lung inflammation, necrotizing cellulitis, respiratory failure. A perfect treatment for this disease would be JAK inhibitors, especially JAK1 inhibitors, which would replace the action of USP18. So our patient here admitted to the ICU at the age of 13 days with severe lung inflammation and interventricular hemorrhage and necrotizing cellulitis, cerebral calcification, and was on high frequency ventilation for months with very high settings. At 7 months of age, he was diagnosed to have USP18 deficiency by whole exome sequencing. And after that, we started him on ruxolitinib, which is JAK1/2 inhibitor, which was never started in the literature in patients in less than school age. At 5 milligram BID, there was no response after 1 week. So we increased the dose to 10 milligram BID, which is usually a dose given to a more than 70 kilograms adult. After a few days after that, he had dramatic response, and he was weaned from ventilation and even oxygen and discharged from the ICU after a couple of weeks, quite dramatically. His lung inflammation was cleared, and the necrotizing cellulitis was healed. And when the dose was decreased to see the minimum controlling dose, his symptoms, he was readmitted to the ICU, and then we quickly increased the dose to the previous one, and he also quickly recovered afterwards. So this patient had a splice site mutation near exon 10, leading to exon skipping. There are other autoinflammatory disorders, which including something like ADA2, defects in interleukin-1 receptor antagonist, and interleukin-36 receptor antagonist. That I will just skip for the sake of time. Then we move to primary immune regulatory disorders, which are inborn errors of immunity resulting from failure to control immune response, usually characterized by lymphoproliferation, inflammation, and organ-specific immunity. And this was discussed nicely by Dr. Mahoud. Several immune pathways, defects may result in immune dysregulation, including different innate and adaptive immunity. This is, as I said, discussed by Dr. Mahoud, the CTLA4 and LRBA role in primary immune regulation and the severe autoimmune diseases that result from their defects and the role of abatacept in controlling the disease. We, in fact, reported the first patient, first family with LRBA deficiency in collaboration with Professor Fawzan Kaya group, and also together with another group from London, simultaneously but independently. This family present with severe GI disease with diarrhea, loss of weight, also recurrent infections, and different autoimmune manifestations. These are biopsies from the lymph nodes show immune lymphoproliferation with increased B cell number, and biopsies from the duodenum and colon show massive lymphocyte infiltration. And here the LRBA protein is absent in those patients. Another example of immune dysregulatory disorder that is not an autoimmune is hemophagocytic lymphohistiocytosis, where there is cytokine storm and overactivation of innate cells, mainly reticular endothelial cells, leading to many inflammatory manifestations, including fever, organomegalies, cytopenias, high ferritin. These are diagnostic criteria of the disease. The patient we described was the first one with familial HLH5, together with the other group of patients in the same publication in collaboration with Professor Genevieve Saint-Cecile from Paris. And our patient found to have defect in MUNC18-2, a protein which is important for the priming of the vesicles that contain perforin and granzymes, which are important for killing the target cells. So mainly a defect in NK or cytotoxic T lymphocytes. Atopic disorders also some of them may be monogenic. And there are several mechanisms that can lead to severe monogenic atopic disorders, involving several pathways, as you can see. So first, defects may involve mast cell signaling and granule disorders, like, for example, mutations in stem cell factor, PLC gamma 2, leading to defect in, and also systemic alpha tryptosinemia, leading to defect in mast cell function, and leading also to increase degranulation of mast cell granules, presenting with urticaria, idiopathic anaphylaxis, and other systemic functional disorders. Other mechanism is also, as you can see, described, some of them described by Dr. Mahawij, is defects in JAK-STAT pathway, gain of function JAK1 STAT5, B, leading to hypereosinophilic, severe hypereosinophilic syndrome, dermatitis, defects in TGF-beta signaling, leading to pathways leading to atopic connective tissue disease, STAT3 gain of function mutation, as previously shown, leading to autosomal dominant hyper IgE syndrome, known previously as Job syndrome, with severe dermatitis, recurrent staph skin infections, and also recurrent pneumonias due to Staph aureus or Aspergillus. So bacterial and fungal infections, and also abnormally blunted inflammatory responses. Similar phenotypes were described with autosomal recessive inheritance due to interleukin-6 receptor, soluble interleukin-6 receptor deficiency, GP130 deficiency, and PGM3, which is important for glycosylation of these proteins. Also, defects in TCR signaling and so-called CBN complex, which is defect in CARD11 or BCL10 or MALT1. We had also actually a patient with MALT1 presenting with severe dermatitis, and recurrent bacterial infection, autoimmunity, and also lymphoma. These are important features that also present in our patient who had MALT1 defect. Actinopathies, genes that control actin cytoskeleton polymerization and related proteins also lead to recurrent infection with skin, food and airway allergies, inflammatory diseases, autoimmunity, increased risk of neoplasia. The famous example is Wiskott-Aldrich syndrome and things related to it, like defect in width and the RPC1B, also DOC8 defects, and more recently Carmel2. They're all defects in cytoskeleton reorganization or actin polymerization. Last but not least, defects in skin barrier leading to, for example, defects in skin SPINK5 leading to Netherton syndrome, defects in filaggrin leading to increased risk of atopic dermatitis, or if it's homozygous, leads to acanthosis vulgaris, and or defects in desmosomes, which are important in adhesions, in tight adhesion between keratinocytes in the skin, will lead to severe atopic dermatitis and metabolic syndrome. So this is the last example that we have of atopic monogenic atopic defects. I hope this made this topic more clear to our audience of and colleagues in the genetic world. And thank you very much for listening and attending.